Hello, future PMI students. My name is Doug Reed, and I'm an Associate Professor of Immunology in the Center for Vaccine Research at the University of Pittsburgh. Although this is the year of COVID, as you can see, I'm in my office and maybe dressed more casually than I would have been before COVID. My research interests focus on the pathogenesis and immunology of infectious diseases, particularly those that enter the body through the respiratory tract. In my lab, we work on pneumonic tularemia, a severe zoonotic disease caused by gram-negative bacterium Francisella tularensis. We want to understand how Francisella causes disease and the role of the immune response in pathogenesis and protection elicited by vaccines. We also partner with a number of labs studying viruses such as highly pathogenic avian influenza, Eastern equine encephalitis, and of course in 2020 we have worked on SARS-CoV-2. I'm going to take you on a short virtual tour through some of what we do. One of our unique abilities is that we are able to infect animals with viruses and bacteria via inhalation. This slide shows you some aspects of that. The picture in the upper left is our Class 3 Biological Safety Cabinet where we do our exposures. It is located in the Regional Biocontainment Laboratory, or RBL for short. The cabinet provides full protection for us during these exposures. Inside this cabinet is a state-of-the-art exposure system which can expose mice, rats, ferrets, rabbits, or monkeys to aerosols containing viruses and bacteria. You can see what that looks like in the upper right, along with an instrument that measures particle size during aerosols. The picture on the lower left shows you a Collison 3-jet nebulizer. This device generates very small particles that reach the deep lung. The picture on the lower right shows you a typical setup for a monkey exposure. In recent years, we have begun to explore aerosol delivery of vaccines and antibodies as well. So one project you could work on in my lab is studying the immune response in the lung after inhalation of a tularemia vaccine. This video shows you an ultrasonic nebulizer generating a large particle aerosol that would land in the upper respiratory tract. Working in the RBL requires special training and protective equipment. Here my lead technician is processing samples from a tularemia study. Another unique aspect of my lab is the ability to use radiotelemetry devices to monitor and record the physiological response to infection in animals. This slide shows you body temperatures of monkeys recorded every 15 minutes for several days after infection with 3 different viruses. We can construct a mathematical model of that data to determine significant deviations in body temperature, which we can then quantify as shown in the table on the lower left. This allows us to compare fever responses in vaccinated animals versus controls. This can tell us if the vaccine was successful in preventing disease, not just death. So Francisella tularensis is interesting to me because it is a facultative intracellular bacterium, which means that it likes to infect the host cells and replicate in the cytosol, much like a virus. The bacteria can cause a severe pneumonia and sepsis, which can be fatal when inhaled, but we really don't understand how the bacterium does this. The host immune response may be partly to blame. We have some preliminary data suggesting that the bacterium can actively suppress the innate immune response during the early part of the infection. So one potential project would be to explore this in further detail to try and understand how the bacterium is affecting the host's immune response to the infection. We are also actively working with collaborators to explore potential vaccines, including live attenuated strains and subunit vaccines. So another potential project would be to study the host B-cell and T-cell responses to the vaccination and how that's able to protect the host when they are challenged with the virulent strains. Outside of work, I enjoy spending time outdoors. Western PA is a beautiful place to live. I love to go camping. This picture shows our teardrop trailer on a trip last year to Little Pine Creek State Park in central PA. Even though I may be getting up there in years, I also still enjoy playing on a competitive soccer team. This picture is from last winter after we won the indoor over 40s Men's Premier League. I may be a little slower and a little more injury prone than I used to be, but I still deeply enjoy playing the beautiful game. I hope you've enjoyed this virtual tour of my lab, that some of the projects I've outlined might be of interest to you, and that we'll see you this fall at Pitt.